Hello world, I'm a protein. Nerve growth factors to be exact, but my friends will call me NGF. I'm responsible for the regulation of growth, maintenance, and survival of certain sensory neurons. I'm derived from the nervous system, right up here in the brain. But because the brain controls everything, I'm used throughout all the body's systems. An example of this is immunity. During inflammation, I am released in large quantities and increased pain perception underneath the area of inflammation. I can be found on the P13.2 location on the first chromosome, and I'm 52,469 nucleotides long. I started in the nucleus, way back, when I was just a little gene trying to make it in the world. Some RNA polymerase came along and attached itself to 3 nucleotides on my double helix. Held down by transcription factors, my nitrogen bases were unwound and separated. The single strand of nerve growth factor DNA instructions was copied, except for thymine, which got switched out with uracil. Then my genes were reunited and the pre-mRNA floated off into the nucleus to mature. My introns, or information that doesn't code for anything, was removed, and a cap and tail were added for protection outside of the nucleus. As mature mRNA, I was finally ready to go out into the cytoplasm. The process of transcription had finally ended, and I was now ready for translation. The outside of the nucleus is chaos, but my cap and tail protect me from harm. A ribosomal subunit came by and selected a codon, or group of 3. At the same time, tRNA came by, carrying an amino acid. The tRNA selected a codon and translated it to its opposite. This was called an anticodon. The tRNA, now carrying the anticodon, floated off into the cytoplasm, leaving the amino acid. The ribosomal subunit and the tRNA both continue their process down the mRNA strand. The amino acids that were left behind were bound together into a polypeptide chain. I was almost complete, but not quite a protein yet. I had no shape or protein conformation. Combined with other chains of amino acids of many other shapes, like single helix and folds. I had a quaternary protein conformation and a shape finally. I was a real protein and was ready to go out into the body to help. As NGF, I'm needed often, so it was important I got out of the cell ASAP. A vesicle came by surrounding me and brought me to safety to the cell wall. After reaching the cell wall, the vesicle merged with it, and I was released into the nervous system. The vesicle then separated itself from the cell wall and returned to go get more proteins to deliver outside the cell. This process is called exocytosis. So now you know my full story, from transcription to translation, of how I got to be the big bad protein I am today, nerve growth factor.